Hi, my name is kd24388 and my assignment topic is on the expression of EBN A1 protein in mammalian cells to examine its localization within the cell. So my project aim is basically the gene of interest that I've chosen. We're going to express it as a GFP fusion protein, and then we're going to determine its localization. The gene of interest that I've chosen is EBNA1, which synthesizes a protein called the Epstein-Barr virus. This protein is basically very essential for vital viral genome maintenance. So the vector that I've chosen is PEGFPC1. So the reason why I chose this one particularly is because we can visualize the cells under a fluorescent microscope, and it also has a strong CMV promoter for high expression. The next is the cloning strategy. So the first step for the cloning strategy is the vector selection. We're gonna do PCR amplification later. Then we're gonna do verification using gel electrophoresis. Then we're going to do digestion with restriction enzymes such as BAM and BGL, and then we're going to ligate using ligase enzyme. The next step is bacterial transformation. So we're going to use heat shock method. So the first step is to combine the plasmid and the plasmid DNA and the competent cells, and we're going to centrifuge it to ensure thorough mixing, and then we're going to suspend calcium chloride and ligating enzymes, and then we're going to incubate it for 25 minutes, heat shock it for and then incubate it again for some time, and then we're going to add SOC medium, and then we're going to use a shaker to mix it, and we're going to spread it onto the LB ampicillin plates, and then we're going to incubate it overnight. After the overnight incubation, we're going to see growth onto the plate. So the next step is the vector validation method. So we're going to do colony PCR. So in this, we're going to mini prep the plasmids. We're going to select at least 5 colonies from the plate, and then we're going to do restriction enzyme with the previous restriction digestion with the previous enzymes, restriction enzymes, and then we're going to do gel electrophoresis. After the colony PCR, we're going to do Sanger sequencing. This is to ensure there's no mutation, and then we're going to do Western blot, which confirms the EBNA1 and GFP is expressed. And then after the vector validation method is done, we're going to do transfixion into the kd24388 T cells. So we're going to use lipofiction and with the help of we're going to do this with the help of lipofectamine 3000, and then we're going to incubate it for 48 to 72 hours. We're going to visualize this under a fluorescent microscope because we can actually see due to the presence of GFP fused with the DNA. This is my little references, and thank you for listening to me.